Thanks, Cindy, for that whirlwind tour. I'm sure there'll be a lot of questions after the next talk and also due to the next talk and speaker. We have Dr. Sharon Donovan. She is the Melissa M. Knoll Endowed Chair of Nutrition and Health, Professor and Interim Director of the Illinois Transdisciplinary Obesity Prevention Program at University of Illinois, Urbana-Champaign. She is actively involved in her professional societies and served as the 2011-2012 President of the American Society for Nutrition and is currently President-elect of the International Society of Research on Human Milk and Lactation. And Dr. Donovan's laboratory conducts basic and translational research in the area of pediatric nutrition, and she's going to talk on this aspect with the host and microbe interaction. So her title is Non-invasive Methods for Assessing Nutritional Regulation of Neonatal Gut Gene Expression and Host Microbe Interactions.
Okay, well, thank you. We're going to go back to the beginning of the life cycle again, but I hope that I will build on some of the things that Jana talked about and then some of the things that Cindy just talked about. So I'd like to just provide a brief introduction, then we'll talk about a method in my laboratory that, in collaboration with Rob Chapkin, we've been able to non-invasively assess the impact of diet on intestinal epithelial gene expression, and then to relate that to the microbiome. So I always like to start with this quote, and it's actually from an IOM committee, that infancy is a uniquely vulnerable period of rapid growth and development, and as such, feeding changes can have the potential to impart benefit or harm. So I think a lot of the DOHAD really focuses on prenatal programming, but I believe that postnatal programming is very important, and as we'll, I think, learn over time, that probably the development of the microbiome in early life is critically important. And a quote I always like to talk about is that, microbes have been here much longer than we have, so humans evolved within a microbial world. And as was noted, we have 100 times more microbial genes in our body, so we have evolved to them rather than necessarily the opposite. And I think Cindy's point about this loss and when we look at human populations that have more traditional patterns, we see that we're actually losing some of our founder microbes, and it's through sanitation, through cesarean delivery, through formula feeding, and many of the practices that we have today. So again, we know that proper nutrition is critical for health. I like this cartoon, but I can tell you we do not have a nutrition facts label. We're still learning about components in breast milk and particularly what their functions are. Human milk is ideal for infants. It provides all the necessary nutrients, but much of my work really focuses on these bioactive components. So feeding is really not just about nutrition, it's about social and tactile interactions. It also contains many bioactive components. Earlier work in my lab studied IGF-1 in milk, but now we're looking more at factors that stimulate the gut microbiota. We also know from epidemiological and clinical studies that there's health benefits of breastfeeding, and if you look, many of these benefits related to infections, acute infections in early life, ear infections, respiratory tract, GI, but we also see potential for programming of longer-term immune-related diseases such as asthma, atopic dermatitis, inflammatory bowel diseases. So I think the early microbiota is critically important for educating the immune system and to develop tolerance, but also how to mount an appropriate immune response in the face of a bacterial or viral challenge. So we see that there's positive benefits. There also seems to be dosage response, so that when we look at ear infections, about 25% lower risk with any breastfeeding, but 77% lower risk with exclusive breastfeeding. We also see that the genetics comes into roles and has a role, so that when we look at atopic dermatitis, if there's a negative family history, there's not as much protection as if there's a family history. So the link between the mother and the infant and the genetics is very important. So we have a multifactorial fluid Certainly we know that growth differs between breastfed and formula-fed infants, and associations between rapid growth in early life leading to increased risk of obesity has been already mentioned. There's data suggesting improvements in cognitive development. We've really been stuck on IQ, which is about a 6 to 8 point difference. But I think that as we start to look at other behavioral disorders, then we may also see benefits. I'll talk today about the microbiome, but also metabolic programming. So breast milk is more than just providing nutrients to the infant. And we also see as breast milk is important for intestinal development, establishing the microbiome, and immune development. But it's important to keep in mind that these three are continually interacting. We have trillions of bacteria in our gut, which are separated from our bloodstream and our cells by a mucosal mucus barrier and like one epithelial cell thickness. So there's a constant interaction, and there's no reason to think this doesn't, continue throughout life, and is probably increasingly important in the elderly. And one of the organ systems that wasn't talked about today is the gut. And the mouth is the beginning of the gastrointestinal tract, but we do see with the aging, breaking down in barrier function, less acid production. And so I think that we need to think not only about the gut microbiome in elderly, but gut function in the elderly. But keeping in mind that these three processes are closely related to each other. And a lot of what we know about this comes from germ-free animals, so that if we have animals, mice in particular, raised in germ-free environments where they have no microbes, their intestinal development is abnormal, their immune development is abnormal, their cognitive and behavioral development is abnormal as well. They also have increased or decreased risks of a number of diseases. So, the use of a germ-free animal model is helping us really to understand the critical role of the microbiome in health and disease. Many factors influence the development. We used to think that these babies came out of the womb sterile, but we now know by analyzing meconium that there is an amniotic fluid microbiome that the babies swallow, and so there is a microbiome in the meconium, and it does seem to be doing some educating of the immune system. But it's when they come out is when they get, bombarded literally with all the microbes in the environment. What microbes take hold is influenced by host genetics, which we know by twin studies. Whether that infant is preterm or term, but likely a lot of that is how we treat infants in the neonatal intensive care unit, whether they're vaginally or C-section delivered, whether they're given antibiotics, and again, oftentimes these are linked. But now we know that whether there's siblings in the home or pets in the home or smoking or daycare, so beyond that initial colonization, there's a number of other factors. My work in my lab really focuses on nutrition. So if we look at breast milk, now we know the human milk oligosaccharides, which are quite high in concentration and diversity. We also now know that there's a microbiota in human milk. So that's, again, different from infant formula. And the bacterial on the maternal skin. In terms of formula, the type of formula and whether there's pre or probiotics added to the infant formula can affect microbiome. So many of the studies that we have in the microbiome are descriptive, and sometimes they're phenomenon. They're a phenomenon. It's like, we see this microbiome shifts. We feed this diet, the microbiome shifts. But what's really lacking, particularly in human subjects, is the ability to look at potential mechanisms of action. And particularly when you work in neonates, no IRB and no mother is going to let you go in and biopsy their baby's intestine just to see what's going on. So with my colleague Rob Chapkin at Texas A&M, we have applied a non-invasive approach to try to get it, when I talk about looking in the black box of the infant intestine. So this is basically the overall model of what we're looking at, and this will keep me busy for the rest of my career, but we're trying to take components of it. So when we look at what are the components in the diet, and again, our primary comparisons are breast milk and infant formula. And then as we look in the gut, as Cindy mentioned, the sort of metagenome, which is a combination of the microbial genes and the host genes. So what are the bacteria? What are their genes? Most people do this in fecal samples. It's fairly easy to do. What are the human genes involved in the interaction? When we look at diseases, we know that a number of human diseases increase somebody's susceptibility to things like inflammatory bowel diseases or other diseases where they're not appropriately responding to their microbes. Bacterial metabolites, so Cindy mentioned some of the short-chain fatty acids, but there's many other metabolites that are made by the microbes that interact with the host. And the balance of all this is basically influences the balance of health and disease. So to try to get at this side initially, what are the human genes? So as I mentioned, my collaborator, Rob Chapkin, he had developed this method to look at exfoliated epithelial cells. So we've been, most of the data that we have on human infants on the impact of nutrition on gut development comes from babies who've died from SIDS or in car accidents or some other reason that they passed away. And so there's not good data on, their diet and other factors. So he had developed a method to collect mRNA from exfoliated epithelial cells in the stool, and we literally shed about 10 to the 10th of these cells per day as part of our normal gut turnover. So this, again, gives an overview of the two things I'm going to talk about. So the first study was published back in 2010. We took fecal samples from breastfed and formula-fed infants, isolated the mRNA, and at this time we did intestinal gene expression using microarrays. We've now moved on to sequencing. Then in a follow-up study, we had also sequenced the microbiome, both the 16S ribosomal RNA that tells you who's there, and then the DNA or the metagenome, which tells you their genetic potential. And so in a paper published in Genome Biology, we looked at the relationship between the baby gene expression and the microbial gene expression to look at potential crosstalk. We've also been looking at components in the breast milk, but I'm not going to have time to talk about that today, but these are all published. So basically, this was a proof of concept study, so we did not have a lot of infants. We also limited to vaginally delivered term infants because we knew that these cesarean delivery or preterm would impact the microbiota. We also chose second parity mothers because we didn't want to influence their decision to breast or formula feed. All the babies got the same formula, and this was done at a time before prebiotics were added to the Enfamil, so this is without prebiotics. Basically, there was no difference in age, weight, or length of the infants. We also found that the babies grew similarly in these first 3 months, and in both groups, intake per kilogram body weight tended to decrease, but wasn't significant. So we collected stool samples. This was actually done by the mothers in the home, so this is the beauty of the technique, and you can do this at any stage in the life cycle. So we've actually been doing this in toddlers. You could do it in the elderly, and the beauty about it is that most people poop every day or so, so you can collect samples repeatedly. You can get a baseline for one individual, do your dietary manipulation, look at how that changes the microbiota or host gene expression, and then take your diet away. So it allows each subject to be their own control and to do longitudinal studies. So the samples were placed into a trizol reagents, basically to inhibit the RNases, and then the poly-A RNA was isolated through a technique that Rob has. And basically, so mammalian RNA has this poly-A tail on it, and bacterial does not. And he's shown that basically we can get good hybridization of the mammalian RNA from stool, but not the bacterial. So this was applied to an array with 57,000 genes. We found that there were about 4,200 genes that showed a signal, and we limited it to the ones that showed a signal on all arrays. And so we took a very conservative approach because it was discovery and we had a small n. So basically it had to be on all the arrays. So if there were genes that maybe just the breastfed was expressing and not the formula fed, we didn't get this in the initial analysis. So between the two, the breastfed and the formula fed, there are about 1,200 genes that were significantly different after all the corrections for false discovery. We also wanted to be conservative, so we had developed a list of prior knowledge that we hypothesized that these genes might be differentially expressed between breastfed and formula-fed infants. And so then when we queried these genes onto this list, about 20% of them came out as being significantly different. So that's what we focused on. And we did a couple different statistical approaches. One is a technique I'll show you on the next slide called the linear discriminant analysis, was developed by electrical engineers that worked with Rob, who are good at analyzing large data sets. And the other is a standard, what they call Metacore, where you basically put your list of genes into the program and it will put together potentially gene networks or biological pathways. So this is what's called the linear discriminant analysis, and the idea is that here's your line, so there's the linear, and by the computer basically assigning different genes, you look at which genes will allow you to separate out your two groups. So he's done that with colon cancer and healthy tissue. In this case, it was breastfed and formula-fed. So these, again, we look at as potential biomarker genes, and this one EPAS1 seemed to be very significant in being a good classifier for a breastfed infant. One thing to note, the much greater variability among the breastfed and the formula-fed, and this will come out later. So you can do different 2 and 3 gene combinations. These are just looking at some of the genes that were differentially expressed. So they're anywhere from, these were the best classifiers, from 2 to 5.5-fold differential expression between breastfed and formula-fed. And what's interesting is all of the genes were upregulated, and we've seen this now across 3 species, across humans, monkeys, and pigs, that the net effect of formula feeding is to downregulate gene expression in the gut relative to the mother-fed control. And then through the Metacore program, we said, okay, well, what are these genes doing? And what was very surprising to me is the top, the strongest networks were basic signal transduction pathways. So Wnt, Notch are genes expressed in the intestinal stem cells. So Wnt regulates cell proliferation, and Notch helps, and it's a differentiation factor. So I've been a strong proponent of breastfeeding for my whole professional career, but I would never hypothesize that the strongest genes were actually fundamental genes in gut development. So I think when we talk about programming in early life, this is clearly important. TGF-beta also important in tolerance and immune development, but then some that we expected, like cell migration, barrier function, inflammation, were also coming up. So again, we were able to show through this study relationships between diet and host gene expression completely non-invasively in infants. We found some different gene combinations as biomarkers, gives insight into potential mechanisms, but also can represent targets for manipulation by infant formula additives. And then I don't have time to show it, but we've also shown we can do this in preterm infants, and we were able to see developmental differences between preterm and term infants using this approach. So again, we can take it into the NICU and look at a population that's a very high risk for intestinal disease. Okay, so to finish up, I will again kind of hit the high points here. So these were the same infants that we then looked at the microbiota, and we did this at this time as this pyrosequencing. The main point is that you see, just like our gene expression, that the breastfed and formula-fed infants were clustered separately, but you also see much greater variability among the breastfed and the formula-fed infants. And then if we look at particular groups, this is at the phyla level. So again, this is at the highest kind of 40,000-foot flyover look at your microbiota. You see that where infants, both of them were about 55% actinobacteria. This is where bifidobacteria sits. most of us in this room are predominantly firmicutes and Bacteroidetes, but what we found is a very interesting difference, whereas our breastfed infants basically had a lot more Bacteroidetes than the formula-fed. And this type of a ratio has been shown by Jeff Gordon's lab early on to be potentially a very obesogenic type of formula or ratio where your firmicutes are much higher than your Bacteroidetes, so suggesting potentially early life programming of obesogenic pathways as well. So what I showed you were the overall trends, but what we found is that these are, again, your phyla, firmicutes, actinobacteria. So the orange are the breastfeds that seem to kind of look similarly. The green are ones, babies who kind of went to the beat of their own drummer. And these are the formula-fed infants, and I think that Cindy really brought up the importance of diet. So basically, these infants are genetically different, but look at what formula is doing to them. It is driving their, at least at the phyla level, their microbial composition to be very similar. But they're not immunologically similar. So what is the effect of driving them to a common microbiota? So then we had the question, well, is there a relationship between this variation and gene expression? And boy, okay. So we looked at the different genes, and quickly the one category of genes are that function that differed between breastfed and formula-fed were what they call virulence genes. So then we went to basically this was the data that we had from our first study where we knew what the baby genes were. We then did shotgun DNA sequencing of the microbiota, and then with a process called canonical correlation analysis where we took triplets of baby genes and triplets of virulence genes and just queried them against each other. And when we did that process, random genes were not significant, intestinal biology genes were not significant, but what we found was a strong statistically significant relationship between virulence genes in the microbiota and immunity and defense genes in the baby. So it was the first report of really seeing that these virulence genes in the microbes were actually communicating in some way and influence the expression of immunity and defense genes in the baby. So the net effect, so the ones that are down-regulated, basically the net effect was that breastfeeding had a microbiota that down-regulated inflammatory genes in the gut. So the ones that we looked at, some of the cytokines, an LPS binding protein, something involved in selectins, leukotrienes. So there's a number of genes that we saw as a downregulation of inflammation. So even though it was through virulence factors, breastfeeding downregulated inflammation in the gut. So I'm going to kind of basically skip that because I've talked about that already, and I can take questions and just acknowledge primarily the collaboration of Rob Chapkin.